All right, so my name is Cerruti Hooks, and I'm an associate professor and extension specialist at the University of Maryland, Department of Entomology. And today I'm going to talk about one particular project that I have out going on here at the Upper Marlboro Research Facility. I should say this project is collaboration with Dr. Alan Leslie, but he's behind the camera, so I'm going to do all the talking today. So this particular project that I'm going to talk about today is actually part of a multi-institutional project. This project is being headed by Jim Wagenbach at North Carolina State University, and there are several universities across the U.S. who are involved in this project, and we're targeting the brown marmorated stink bug. And overall, the primary objective is just to come up with different, or I should say, better mechanism or protocols to try to manage the brown marmorated stink bug. The particular project, or I should say, sub-objective that I'm involved in is centered around conservation biocontrol or habitat manipulations. And that sub-objective, we're trying to look at different mechanisms where we can enhance the natural enemies of the brown marmorated stink bug. In this particular project here, we're looking at marigold. One of the marigolds species that we like to look at, or cultivar, I should say, is single gold. And one of the things we determined about single gold is that it does a good job of attracting natural enemies, particularly parasitoids. So we felt that it would be could serve as a good insectary plant to attract natural enemies that would also use the brown marrow stink bugs, mainly their eggs, as host or prey. So let me talk a little bit about the sampling protocol that we do. I mean, I'll start with how we look for stink bug egg mortality. And we basically look in the undersurface of the leaf. Once we find the egg, there are a couple of categories that we can place that egg in. And again, the whole idea is to determine the mortality. So we look at the percentage of them that hatch, percentage of them that are eaten by predators, and we separate chewing predators from sucking predators. We look at those that are parasitized, percentage that are parasitized, and we also look at percentage that don't hatch and what we call unknown mortality. And unknown mortality is when an egg doesn't hatch, but there's no indication of predation, whether it's chewing or sucking predators, and there's no indication that it was parasitized. We also have a one that's called missing, and missing is when we find an egg and we go back to look for that egg and it disappeared. And of course, we look at the percentage that also reach that first instar stage. Because once they reach the first instar stage, there's less natural enemies that are out there that can successfully attack it. The other thing we do is, of course, we're trying to see what natural enemies are responsible for that, and they're monitor arthropods as well as natural enemies within the edamame. We do sweeping. We do that once a week. And we also use these yellow sticky cards. And these yellow sticky cards are mainly to monitor the different families of parasitoids that we have visiting the plots. And I should mention that we not only look at the brown marrow stink bugs, but we also look at some of the local stink bugs. Because one of the things we have determined in that looking at egg mortality of brown marrow stink bugs is that the natural enemies, and particularly predators, tend to do a good job in their control. But we found that there is one local stink bug, the green stink bug, that seemed to escape both predation as well as parasitism. So we were particularly interested in how surrounding edamame, which is vegetable soybeans, by this marigold plant would impact parasitism of green stink bug eggs. And again, going back to our initial findings were that we could enhance mortality of stink bug eggs. Mainly it was through greater parasitisms. We also saw that that was the same with the green stink bug eggs, which we were happy to see. So this is our second year, or I should say third year. The second year was we sort of ran into a mishap when some deers came in and ate up the plants. So this is our second year, and we're hoping that we can have stink bug eggs population high enough that we can see if we can repeat the results that we saw the initial year. Beyond that, what we would like to do is start also looking at the marigold in other systems. Next year, I would like to move to sweet corn, and one of the reasons I would like to move to sweet corn is because, of course, it grows a lot different than edamame, so it'll give us a chance to look at the potential use of marigold in a plant that is totally different from edamame. We did a similar study in field corn where we looked at insectary plant, and the problem that we found with the insectary plant was that it worked really well, that it attracted a lot of natural enemies, but it served as a sink. In other words, the natural enemy would not move into the cropping system. So we think with marigold, it may be one of those plants that's not going to attract a lot of herbivores. So in other words, the natural enemies that are coming there, that basically come in there to feed on the nectar, and then they will move into the plant system. Um, in this case, edamame and parasitize those stink bug eggs or other herbivores that are in there that are doing damage. What I can say in conclusion is that although we do find that this marigold tends to work well, so far, at least from our initial study, we're not sure if this is how farmers would plant the marigold seeds. Of course, we do it from transplant. If we determine to find that it is effective consistently, how would farmers go about, especially those who have large acreage? What would be the cost of them trying to um, put a border of marigold around their fields?